welcome to Science Friday.
Here's some for E as well. The one on the top here is one of the ways that you can define E.
What makes a field more likely to have an unbalanced gender distribution?
So this can be, like, in the case of this paper, gyrification or brain folds.
A neuroscientist started to look at the brain
computers, like the math
more as an
computers,
interconnected
I'm
are very
incredibly negative
area.
I'm an 11, and I'm going to be presenting about cancer prevention, specifically in blind mole rats. But before I get into my presentation, I want to give a really, really big shout out to the data team that helped me throughout this process. You guys took me from 0 to where I am now, and I could not be more grateful for so much fun. I'm really excited to present. So with that, why talk about this? For me, about a year ago, I had an ovarian cyst. It's called a cystadenoma, and it's 3 centimeters long, which is about a foot. It took up my whole abdomen, and I had to get surgery to remove it, which is kind of crazy. Of cells. And cancer sucks. Like, it is really hard to get diagnosed. It's really hard to have to go through that and go through chemotherapy or radiation. 1.6 million people, over 1.6 diagnosed with cancer every year. And a lot of, like, why do we not know why this happens? What happened to me biologically, like in my body? And why has this been around for so long and we don't know how to prevent it? So I first want to start by talking about paradox, which is the idea that when an animal, like the bigger an animal is and the longer it lives, the more likely it is to get cancer. And this makes, like, this makes a little bit of sense, right? More cells, more opportunity. But this actually isn't the case all the time. Why? Because certain animals have anti-cancer mechanisms in their body that make it so that they are really good at preventing it. Which leads me to blind mole rats. Blind mole rats, they live for 30 years, which is a really long time. Researchers have never observed cancer in a blind mole rat. I mean, like, that's just kind of crazy in itself. I mean, if you think about dogs, unfortunately, dogs get cancer all the time, and they live for about half as long as mole rats do. Why do blind mole rats, like, why are they really good at preventing cancer in their own bodies? Well, the idea is there's this enzyme called DNMT1, which leads to DNA methylation, which is essentially where your DNA goes like bloop, and it can't be read. It gets all tingly-tangly. And DNA methylation leads to RTE expression. So what is RTE expression? RTE expression is this little DNA, and it pops out of its DNA sequence and pops back into another genome. And so this can kind of be really bad because let's say you have a fat receptor genome, and you have a piece of DNA popped out and popped into that genome for the fat receptor. That genome is no longer making a fat receptor. And so without the fat receptor, your body thinks that there's no fat. Similar thing could happen with cells. So you have the DNMT1, decreases DNA methylation, or sorry. DNMT1 causes DNA methylation, decreases RTE expression because this can't be read, right? So the idea that this paper is proposing is that while all of this is true, this is the pathway to connect it to this. But so why do we care about RTE? Because DNMT1 leads to DNA methylation, which leads to RTE expression, and RTE expression somehow leads to tumor cell death. Why do we care about that? Well, and how do they figure out that connection? I'll start by explaining how blind mole rats play into this. Blind mole rats are observed to have really low levels of DNMT1, the enzyme. And with low levels of DNMT1, they have less and more RTE expression, which then leads to tumor cell death. But is it the RTEs jumping out, popping out of that genome and into another one that's causing the tumor cell death, or is it something else? And why do we care about these in-between steps? If you exercise, you're exercising, something happens, something else happens, and then you get muscles, right? But what if you were able to isolate the step right before the muscle, and then you were able to get swole? That would be cool. And the same thing applies here. So if we know that RTE expression leads to something which leads to tumor cell death, if we were able to figure out what that is and isolate the step right before, we would be able to cause tumor cell death without all of this. So how did they figure out that RTE expression leads to cytoplasmic DNA? This is the important part. That cytoplasmic leads to this pathway called C-gas sting IF, which then leads to tumor cell death. How do they do this? Oh, here's where the chair comes in. Okay. So we have blind mole rat cells. And in these cells, they dyed the sting dimer, which is the measurement by proxy for the activation of the C-gas sting pathway. And what we found is in these two drugs, abacavir and zidovudine, which are anti-HIV drugs, but they also happen to decrease the levels of cytoplasmic DNA. When you have less cytoplasmic DNA, we find that sting is not activated. It's not there. But when we do have cytoplasmic DNA, which is the one without the drugs, the control, we see that sting is activated. That's what the dark spot means. And so what does this mean in terms of cytoplasmic DNA leading to C-gas sting, and how do we know that all of that leads to tumor cell death? Okay, in this graph, in our abacavir and zidovudine, the blue and red, you can see that it increases and increases. Population doubling is just another way of saying the amount of cells. So it's increasing and it's increasing, and it keeps increasing. And remember that these two are the ones that don't have any of this. But when in our control, when we do have cytoplasmic DNA, the cytoplasmic DNA, or the cells or the population is increasing, and it's increasing, and then it decreases and stops. So cell death has occurred when cytoplasmic DNA is present, and therefore the C-gas sting is activated. All of this leads to tumor cell death, which is pretty cool, because now we know where the RTE expression comes, where that leads to, and how that can connect to cell death. And with this, we can isolate the activation of this pathway, and that can lead to tumor cell death. But this isn't all we have to figure out with this paper. We also have to figure out how low levels of DNMT1 really block cancer cell growth. So what figure C is showing is you have SH-DNMT1 and SH-scramble. SH-DNMT1 is another way of saying you're knocking down DNMT1, which means that DNMT1, the protein, is not being produced. So this, no DNMT1. But in SH-scramble, you're knocking down scrambled letters. So you're not knocking down DNMT1, and DNMT1 is present. And what do we see with this? Well, over here on this graph, remember, DNMT1 not present, DNMT1 present. We're seeing a large decrease in the relative colony area, or in other words, the amount of colony that is covered by cancer cells, those purple dots. But another really cool part about this is that HeLa cells, the cancer cells that are being tested with this, are human cancer cells. So this mechanism can also work in humans. So why do we care about this? Why does this matter to talk about? And what's the deal? Well, something that's really interesting is that because we can use this in humans, we don't have to go through all of the steps of the poisoning your bodies with chemoradiation or chemotherapy and radiation. And it's introducing a new way to prevent cancer in bodies that isn't just hoping that all of the tumor cells are going to die first. So instead, we have something that kind of gives hope for the future in terms of how we can develop ways to prevent cancer. And so I'm hoping that with the research that has been done with this, we can essentially figure out other ways to cure cancer that aren't as bad for humans and their bodies as a whole. And blind mole rats are pretty cool. Thank you guys, I'm here for questions.